What's going on, everybody? Welcome back. Today we are going to be doing part 2 of the IAT Bootcamp, and we're going to be talking about cellular reproduction. That's going to include the cell cycle, mitosis, and histology. So let's get into it. So, the cell cycle. The cell cycle is basically just the entire lifespan of a cell, and you can kind of see that illustrated by this diagram over here, but we're going to talk about this a lot more and go into more detail on it. So, overall, there are two main phases of the cell cycle, those being interphase, which takes up 95% of the cell cycle, and the mitotic phase, which takes up 5%. So interphase, again, is going to be taking up the most time, as you can see from this diagram. This isn't really greatly drawn to scale. Obviously, it kind of looks like a 75-25 split here, when in reality, this blue arrow should cover just about the entire circle. And the ultimate purpose of the cell cycle is to duplicate the cell, right? So we're going to be talking about how interphase basically prepares the cell to replicate itself, and then mitotic phase is where that replication actually occurs. So the first part of the cell cycle that we're going to be talking about is interphase. Like we said, interphase takes up a vast majority of the cell cycle, and interphase itself can be split into three different phases. The first one is gap phase 1, the second is synthesis phase, and the third is gap phase 2. In gap phase 1, the cell is going to be just growing larger in general and is going to be preparing to duplicate the genome. What is the genome? The genome is all the DNA for an organism. This preparation is important because in order to duplicate the cell, which is the entire purpose of the cell cycle, we need to duplicate the DNA, because the DNA is what is like, it's all the important stuff in the cell, right? It's what makes the cell, what makes it do its actions and whatnot. So yeah, that's gap phase 1. Synthesis phase is where the actual DNA replication is going to occur. So gap phase 1, we're preparing to duplicate, and in synthesis, we are actually replicating the DNA inside of the cell. And finally, gap phase 2, the cell is going to review the DNA that it just replicated. So this is basically looking for errors in replication, and this is ultimately going to be preparing for mitosis. So again, these three phases take up 95% of the cell cycle, and they are basically all leading up to mitosis, which is where all of these prerequisites kind of come together and allow the cell to duplicate. All right, so next, now that we've talked about interphase, we can talk about the mitotic phase. The mitotic phase can be split into two parts: mitosis, which is a majority of it, and cytokinesis. Cytokinesis basically overlies part of mitosis, but it's important to separate the two because there are just two different terms that you have to know for an anaphase, right? And mitosis is going to be the division of the nucleus. So when we're talking about mitosis, it is how the nucleus is being divided, whereas cytokinesis is focusing on the division of the cytoplasm. If you're not familiar with like the structure of a cell, this image right here shows the nucleus obviously is just this dot here. So in mitosis, we're going to be talking about how this nucleus is being separated into two. Whereas in cytokinesis, we're talking about the division of the cytoplasm. So this whole surrounding area basically that is contained by the cell membrane, we're going to be talking about how that is split into two different cells. And one thing I forgot to mention here is that obviously the four parts of mitosis, we have prophase, metaphase, anaphase, and telophase. And in the next slide here, we're going to break down all of those and see kind of what happens in all those different phases. So let's break down all these stages of mitosis. First, the first phase is going to be prophase. In prophase, we're going to see two main things: the dissolving of the nuclear membrane and chromatin being turned into chromosomes. So first of all, let's talk about the nuclear membrane. The nuclear membrane is simply this circle around the cell. So this is our nucleus, and it contains the chromosomes / chromatin. And basically, we're going to see the membrane around this begin to dissolve, allowing the chromosomes to be kind of like free-floating in the cell. And for chromatin turning into chromosomes, our chromatin are just going to be these kind of like macaroni noodle looking shaped things in here, the dark pink things. And those are essentially going to be turned into chromosomes, which we can see in the following phases are kind of these more skinny little macaroni noodles per se. So yeah, prophase, we have these two things happening, and then we'll move on to metaphase. In metaphase, the chromosomes, so again, chromatin over here, now we are chromosomes. Now these little pink things are chromosomes. The chromosomes are going to align along the midline of the cell, which is right around here, and they're going to attach to the mitotic spindles, which are going to be these little spindles or just lines that are attaching to the chromosomes. And the chromosomes have also been replicated at this stage, so they are ready to be separated. And when we see, you can't really see here because I drew over it very well. Let's see if I can erase it. Yeah, so the chromosomes here, you can see they're kind of in pairs. They're making little X's. And these pairs are called sister chromatids, right? So the DNA has been replicated already, if we remember from back in interphase. So the DNA has been replicated. So we have these sister chromatids, and they're essentially two of the same thing. So they're ready to be separated. So now in anaphase, we're going to see these chromosomes being pulled apart to the opposite ends of the cells. So again, we see kind of some separation now between these sister chromatids, which are now becoming their own chromosomes because they are separated. They're no longer sisters because they're not together. Next, telophase, we're going to see the formation of a new nuclear envelope, which you don't really see in this diagram because telophase is just kind of the beginning of that step. And we're also going to see the chromosomes being turned back into chromatin. Again, you don't really see that very well in this diagram, but just know the chromosomes are being turned back to the chromatin like they were initially here in prophase. And another notable thing too about telophase is that we just have more separation between the opposing chromosomes. And finally, cytokinesis. Cytokinesis is actually going to overlap number 3 and number 4, so anaphase and telophase. This is going to be the division of one large cell cytoplasm into two smaller cells. If you remember on the previous slide, we talked about how cytokinesis is referring to the separation of the cytoplasm, which is all this open space in the cells, whereas mitosis is the separation of the nucleus. So by the time we finish telophase, the nucleus has been separated, so now we have to finish it out with cytokinesis where we are separating the cytoplasm. And the main indication of cytokinesis, which again, remember, is occurring in anaphase and telophase, is the cleavage furrow. What is the cleavage furrow? That is simply this little dip or this like separation of the circles that you can see beginning to form in anaphase, telophase, and then by cytokinesis, they fully kind of pinch off and form their own separate cells. So now that we have an understanding of all these stages of mitosis, let's look at how you're going to have to be able to identify these in an actual histological slide. So obviously this is a diagram. We're going to have to be able to identify this in a microscope slide. So let's look at this next slide going over that. So now that we have talked about all those phases on the diagrams, here we have all the phases of the cell cycle, except for these are actually the histological slides that we're going to have to be able to identify. So first of all, interphase, we're just seeing a normal cell, right? We have our nucleus, we have our cytoplasm. Nothing much is really happening here. So you probably wouldn't be asked to identify this as like what phase of the cell cycle this is, because this is just a plain old cell, right? So not a lot happening here. Now, moving on to mitosis, our first phase of mitosis, prophase, we remember that we have the nuclear envelope developing, and we can see that where in interphase we have this pretty defined nuclear envelope around the nucleus, but in prophase it's not there, right? We just see all of the contents of the nucleus, which are the chromatin, which are turning into chromosomes, if you remember that. So yeah, that's how we would kind of identify prophase. I am going to go over in the next slide as well, just kind of like exactly what you're going to be looking for when you see a slide to identify each page. So this is just a brief overview. Next, metaphase, we again see the chromosomes lining up on that midline, and we can also kind of see these mitotic spindles beginning to form. So that's going to be kind of the main parts of metaphase that we're going to be looking for. And then anaphase, we see the chromosomes being pulled apart by the mitotic spindles towards the opposing ends of the cells. And then telophase, we see this pulling just kind of continue, and we can see that cleavage furrow developing even more, which, remember, that is our sign of cytokinesis. And cytokinesis is occurring during anaphase, telophase, and then obviously it finishes kind of right at the end of telophase. So again, telophase, we see the chromosomes being pulled further and further apart. The mitotic spindles are kind of becoming less and less apparent, as you can see here, as opposed to an anaphase where they're pretty obvious. There's these obvious lines kind of pulling between the chromosomes. And yeah, the cleavage furrow just kind of gets more and more developed as we go from anaphase to telophase, and then eventually cytokinesis, we have a whole new cell that is formed. So let's go over exactly what we should be looking for when we see a histological slide and we are asked to identify what phase of the cell cycle we're looking at. All right, so these are going to be the keys to identifying the phases of mitosis in histology. So for prophase, we're going to be looking for squiggly lines. What does that mean exactly? We're looking for, when we see squiggly lines, that means the nuclear envelope has been dissolved, and we are kind of seeing the chromatin being turned into the chromosomes. So again, we can look back at this previous slide where we see all these squiggly lines just kind of sitting in here, and there's no defined nuclear envelope like we have in interphase. So that is going to be the main way we're going to identify prophase when we are looking at a slide in a microscope, is by the squiggly lines, which represent our chromatin/chromosomes. Next, for metaphase, we have a few more criteria that we're going to have to look at. So we first want to see the chromosomes aligned along the midline of the cell. This is going to be pretty unique to metaphase. Sometimes you can see this in very early anaphase if the chromosomes haven't been pulled apart very much, but a pretty sure sign to identify metaphase, again, is that we have these chromosomes lined up and touching on the midline. We're also going to see the mitotic spindles are now visible. So again, prophase, we don't see any mitotic spindles, but metaphase, we do. We see them, these lines kind of pulling back here and touching the chromosomes aligned along the midline. And also the chromosomes, again, are still close together. This is just kind of a way to compare metaphase to anaphase and telophase, because we can see the chromosomes just kind of continually get pulled further and further apart, but in metaphase they are very close together, which is kind of a key defining characteristic. Next, for anaphase, we want to see the chromosomes being pulled apart. So we want to see some slight separation, not the major separation we see in telophase. So we can see here we have a little bit of separation being formed between these two darker spots, which are our chromosomes. And we also want to see the mitotic spindles are still very apparent, right? So again, comparing this to telophase, we see that the mitotic spindles are kind of starting to disappear, right? They're not very apparent. As opposed to anaphase, we can see these long streaking lines between all the chromosomes, so we can very clearly see our mitotic spindles still. And finally, we want to see no obvious cleavage furrow. So if you remember, anaphase and telophase fall under cytokinesis, right? So the sign of cytokinesis happening is that we have the cleavage furrow forming. However, in anaphase, we don't want to see a very large cleavage furrow, because that is only going to occur in telophase and in cytokinesis, which is kind of after telophase. So we can see in this anaphase, there's not really a largely defined cleavage furrow. You could make an argument there's one maybe right around there or right here, but again, it is not super apparent, which is another good sign that we are in anaphase. And finally, for telophase, we want to see the chromosomes that are far apart. So comparing telophase here to metaphase and anaphase, we can see there's a good distance between these chromosomes as opposed to the previous two. Next, we want to see, again, like I mentioned before, that the mitotic spindles are starting to fade. We can see here the chromosomes are far apart, and that we don't really see any defined lines of our mitotic spindles anymore, as opposed to anaphase and metaphase, where we see these really defined pulling lines. And finally, we want to see a clear cleavage furrow and divide between the cytoplasms beginning to form. So again, this is something I mentioned before. We have the very clear and obvious cleavage furrow, right? This kind of pinching off that is occurring between the two cells. And additionally, we can even see a separation of the cytoplasms beginning to form. This is a sign of kind of late cytokinesis, which is going to overlap with telophase. So that can be a little bit confusing. Like if you were asked to identify cytokinesis, you would likely be also being asked to identify cytokinesis out of anaphase and telophase. So just be careful when answering questions like this. Ensure that, are you supposed to be answering cytokinesis, or are you supposed to be answering a phase of mitosis? So obviously you can get tricked pretty easily on questions like that. So just make sure you're being diligent, pay attention, and just remember those criteria that we're talking about here. I highly encourage you to put these in some flashcards and just study them. Use verbal active recall like I've mentioned on my other social pages, and just get these down so that you can recall these characteristics consistently. Alright, that is going to be it for this video covering the cell cycle. If you guys have any questions, if I covered anything too fast, please let me know. Drop a comment on the video or just shoot me a DM on any of my socials. I'd be happy to explain anything that I talk about in this video. And yeah, I hope you guys learned something.